This is a production of Cornell University.
Here today, Moira.
Well, thank you. It's my pleasure to be here, and it's also potentially a little bit of a sad one because this may be the last seminar I give as a Cornell employee. But we'll get to that at the end. Don't want to have too many spoilers. So today we're going to start off with some questions. What are specialty crops, and why should you care about them? And what would be a specialty livestock? And then we'll also talk a little bit about why technology adoption is so hard to do in applied breeding. And then if you're not familiar with what Breeding Insight is, I'll give you a little background on what we do and how we got started. But the most time we're going to spend on all of these successes BI has had while at Cornell these past 6 years. So that is the most of the talk here. And we'll touch a little bit on our future going to University of Florida, Gainesville as our new future home at the end. So the reason you should care about specialty crops is they're in your daily life. They are the things that you go to the grocery store to buy, either as produce or juiced or fresh, frozen, dried, any number of things. You can find them everywhere, and they're in your diet. And specialty specialty crops aren't just for nutrition. There's a lot of the things we do that are culturally significant to us that we are really considered specialty crops, like chocolate, coffee, sugarcane, of course, wine from wine grapes, tea, and vanilla. All things we really enjoy in our cultural experience, but maybe don't really provide a lot of nutrition. And specialty animals, you probably have some of these things that you encounter at least weekly in your life. So things like aquaculture for either mussels or fish, food fish. Lots of mammals fall into this category. Alpacas, goats and sheep, Angora rabbits or meat rabbits, and even bison and dogs. There are some bird species like the large flightless birds that people raise for meat, and also quail. People like to have a lot of quail in their backyards these days, and insects too. So the most common one you're probably familiar with is honey. That comes from honeybees, but people do raise crickets for other kinds of feed, and of course silk moths produce silk for fibers. The reason you really should care about these things is because even though non-specialty crops is about $135 billion last year in cash receipts, all of the specialty crops combined is about 108 billion. And if you look at livestock, it's not quite as equally divided. So most of the cash receipts for livestock is coming from major crops or major livestock, like eggs, poultry, hogs, and of course cattle. But the specialty livestock is nothing to sneeze at. It's still $8 billion. And this covers what we talked about on the last slide, but it also includes other uses like meat, dairy, animal feed, and fiber. So specialty species are essential for human nutrition, and they comprise a significant portion of the American agricultural income of about $116 billion last year. So consider this as we talk about securing the US food sources, that those strategies really need to include specialty species. This is where your nutrients are coming from, your vegetables, your fiber. These are things that enhance your living and your health. So we'll talk a little bit about why technology adoption is so hard. And it really just stems because we always think of things like, well, if we do this in corn, it'll trickle down to something else, right? Well, if you take corn and soybean on the left here, and you think about what it takes to go from seed to seed in that season, you're looking at a lot of fully mechanized services that are happening. You can have low labor costs because you can have one person drive a tractor over hundreds of acres. They're annual crops, so there's a very short window they're all working in, so they don't have a long longevity in the field. They act as functional diploids. They're seed propagated. There's a lot of commercial breeding happening. They have very high yields, and the harvest is dry, so you can store it longer. But if you look at blueberry and alfalfa on the other side, there's a little bit of commercial breeding. And so that means that a lot of the other things that are technologically based in corn and soybean just don't transfer very well to something like blueberry or even alfalfa. And it's compounded because breeding is actually a lot harder to do with the specialty species. They have much greater logistical and financial constraints than major crops and livestock. Their biological challenges are more diverse. They have different, their environmental limitations. There's things they can't do in a greenhouse. They have to do it in a large field. And then there's a lot of technical barriers. A lot of the know-how you might want in your program may not be available to you when you need it. And so when we think about someone who is breeding in a specialty a realm, they're often having to wear multiple hats. They're having to manage the sample collection and the trait data collection and making sure that the trait data collection tools are functional before you go out. Managing the breeding program from year to year, managing the event documentation, like when is fertigation happening, when is pruning happening, being aware of the stakeholders' needs for new native traits, so they're tapping into the germplasm, the MPGS germplasm banks. They're storing genetic data, making genomic analyses, and then doing a lot of decision support and reporting. And that means they're wearing multiple hats the whole time they're doing this, and they're not under infinite time. So these are highly constrained time periods where they're having to do everything to the best of their ability and then bring all that data together to make decisions. So I'm going to start by introducing you to somebody who was really pivotal to getting Breeding Insights started in the first place. So this is Chad Finn. He is, this is in 2019. We met him as our first breeder meeting we went to. And unfortunately, he passed 4 months after this photograph was taken suddenly. But Chad Finn released or co-released 51 berry cultivars that are still grown all over the world. So he's not just working one crop, he's working in 4 crops. And he relied on only one piece of software, Excel, to bring together all these years worth of performance data to make selections across these multiple crops. And he only used phenotypic selection over his 26-year career, and most of his decisions, he would tell you himself, are based on intuition and experience. If you've got 26 years to do that, you can be successful too. If you don't, though, and you're like most modern breeders, you may require more sophisticated data management and analytical tools than Excel can offer you. You're doing a lot of data collection all throughout your field season, and then you're also having to recycle and integrate that data into the next year's cycle and on and on. So plant breeding and animal breeding really are fields of data science where you're bringing together knowledge from very large, disparate, complex data sets together using statistics, mathematics, AI to really understand what that data is telling you to make a decision that is actionable in your breeding program, whether it be selection, culling, or maybe optimizing mate pairs. So Breeding Insight was really founded to kind of bridge this gap that we were talking about. Technology integration is a long-term thing, and to get adoption to hold, you really have to immerse it into the program that it is being used in. And it's risky for a breeder to pick this up. And it's time consuming. They're already doing everything else. You're going to throw something new at them, they're just not going to have time for it. So they really need support to do this. So we provide low-risk tools, software, and a holistic support system to enhance those specialty breeding programs to really make sure that we get durable technology adoption. So our mission really is to transform breeding by the implementation of genomic insight and selection as part of routine breeding across all of specialty crop and livestock breeding across USDA ARS. We help connect and build resources for specialty crop and livestock breeders. We are a USDA ARS funded initiative currently located at Cornell, footnote. We are a service and technology transfer model that provides additional support to USDA specialty programs, but we also work with their university partners whenever it's appropriate. And we're mostly a team made up of scientists and software developers. We're not a private company yet, We are not interested in duplicating established technologies, techniques, or patents. And we don't sell or receive any commission for any technology that we provide or recommend. If we're recommending it to somebody, it's because we have really good use cases to support that it would be a useful addition to their program. And as Mike said, that the inception for Breeding Insight really happened in 2018, but we really didn't start working until 2019, and we started with 6 species. Pretty diverse group of species to start with. Those 6 species actually covered 15 different breeding programs at USDA in different areas for different market types. In 2022, we took on an additional 10 species and doubled the number of breeders we were supporting. And then in 2024, we took on another 11 species and now have more than 50 breeding programs that we're supporting. And we work with a hub and spoke model. So a lot of our core resources are here at Cornell, and we work by distributing support out to those locations as needed. And oftentimes we can go to some locations where we have many of our crops and do kind of a one-stop visit there. So the next section I really want to kind of highlight is we've done a lot of work at Cornell for the past 6 years, and it's been a great place for BI to start out and build success. And it's been influenced by a lot of people that aren't even in this room. So I'll start by saying that we really try to provide tools at every level, and we're looking to identify the most common solution for all species. We want to build robust tools, techniques, and solutions that work across ploidy levels, breeding program sizes, any kind of biology system, and whatever breeding techniques the breeder is using. So even though we support each of the species individually, a lot of the services are spread across all of those. So we'll start with genotyping. This is just a quick overview of all the genomic complexity that we have that right now at the species we cover. We have a haplodiploid system. We have a lot of diploids, but some of them actually have sex chromosomes, and then some actually use a 3-gender system for their breeding program. And then we have the full range polyploids here, from 4x to 12x. And a lot of these are autopolyploids, which have just been amazing to work with, but they've been very difficult to get started with. And we really wanted to find a solution that fit as many of these use cases as we could at one time. And we did. So we started partnering with Diversity Arrays Technology in Australia, and they have been actually one of the best partners we've ever had. They're very flexible to work with. And we worked out a workflow where the breeders can collect the tissue at their location. They send it to DART for DNA extraction and genotyping. They send the data to us and the breeder, and we help do the analysis at the other end. And the nice thing for the breeder is they do the tissue collection, and then they can go do something else. They're not having to wait around and do the DNA extraction themselves, or it's out of their hand. They've outsourced it. It's really useful for them. And to date, we have genotyped over 90,000 samples, and we've discovered over 160,000 haplotypes using these marker platforms. And so we start by building, for most of the species that don't already have a fixed marker panel, a 3K DART tag marker panel. And you can see the ones we've already developed are over here, but we've got almost an equal number that are still in the work or are in various stages of testing. And the benefit of this is that for each of these species, they wouldn't have enough buying power to get a company to pay attention to their specific needs. But because BI is the wholesaler of this organization of all of these crops together, we effectively become a better advocate for all of the crops to DART because we are a large provider of service. We pay for a lot of the services. So I think that relationship has been good for both DART and for BI and for the species involved. And so next I want to kind of talk about what's so special about the DART tag platform. And I'm going to use 2 examples from 2 autopolyploid species, so alfalfa on the top and blueberry on the bottom. And let me see if I can get this thing to work. Okay, is that working? Okay. So unlike most SNP arrays where you're looking at a single target site at each locus, The beauty of the dart tag system is it's using a single primer to create an amplicon of either 54 or 81 or 109 base pairs. And because you're using a single primer, you only have to bind on one side of your SNP. And so you read through the other side and you can actually detect more off-target SNPs than you would have found just through your discovery. So if you were on a SNP array, this looks like an AT SNP, but when you actually look at all of the alleles we've seen, you can actually see that there's, what, 8, 9 alleles here? So instead of just subtracting down and saying it's A or T, now we have 9 alleles we can actually put into the genomics model and ask which one of these is giving the highest genetic signal for the trait we care about. The other good thing is that for autopolyploids, this really improves our phasing for linkage mapping. And it improves our dosage calling for those polyploids as well. So we've been collecting all of these microhaplotypes. We're assigning fixed allele IDs so that they can be publicly shared and utilized together in the community. And we're using these microhaplotypes in downstream genomic analyses. And one example of how we're using this is just shown in these upset plots on the right here. And so this is looking at the frequency of common, rare, and private alleles in either a repository on the top here in alfalfa, or by breeder in the bottom example. So if you look down here, there are 4 lines here. These are 4 breeders. Their initials are just shown. You can see the number of alleles that are common across 3 or more breeders, some that are rare, only 2 breeders out of 4 have it, or are private. So this allows those breeders to actually manage their germplasm better, and it improves the ability to share your germplasm with other people. You have a lot more insight into what you're actually sharing. Next, I want to talk about bioinformatics, and I'm just going to talk about one example. So this is our Big App and Big R apps. These are low-code, no-code tools that we've built to bridge the gap between breeders and the genomics computational tools that we use, or a lot of people are using as scripts. So it's a web-based application that performs genomic and bioinformatic analyses. And again, it's meant to be used as it's fully functional as a no-code solution. So could be used with undergrads, capstone projects. It has a lot of tutorials and help pages, and it has broad species support across ploidy. And a little bit more about it is that there are a lot of analyses you can do, and the beauty of it is you can pop in and out. You don't have to go through a full workflow. You're not trapped in it. If you want to start with your VCF and come in and do some filtering and then get back out into R and do whatever you want, then this makes it easy to do that. But you can also do summary metrics. So you could look at MAF, or you could be looking at heterozygosity levels. And then there's some population structure tools in there, DAPC and PCA, and GWAS and GS support in there as well. It also produces publication quality figures. So one of the goals I would like to do when I nominally move to Florida is build a capstone undergraduate project course that allows students to use this and pairs them with public scientists that would like to have help analyzing their genomic data and analyses. Going next to phenotyping. Phenotyping is a little bit different than genotyping. There isn't one solution that fits everyone. So we really kind of have to find the economy of scale of what we build in a different way. So for example, if you look at, we did a project with the Geneva hemp collection up in Jack Stansel up in Geneva, and they were looking at doing some plant characterization data, and they really wanted to just take the pictures with Fieldbook in the field and then count serrations or get other phenotypic data out for GRIN. So the images were collected on a pre-made palette with size and orientation markers and a QR code for the accession. The image was auto-oriented and corrected during pre-processing, and then the leaf attributes, including number of serrations, were output. In potato, they were looking at storage defects and other kinds of production defects. These would be things like knobs, growth cracks, sprouting, or greening. And these potatoes are just running through a conveyor belt under a mounted camera system. And then we're using AI and machine learning to actually do on-the-fly detection of those prevalence of these different defects. And the other benefit of working in BI is you get some weird projects that you would never get as a plant biologist. And this is one of my favorites because this one, I would have, if somebody had told me we were going to do this, I would have been like, you're out of your mind. There's no way we're going to do this. But I was wrong. And I want to kind of talk you through what you're looking at here, because I'm sure you guys don't look at salmon eggs all the time either. So in the raw image, our salmon breeder said, you know, I can see that there's some blanks here. And we're like, okay, well, what's a blank? Well, these are eyed eggs, so they're fertilized, but there are some eggs in here that look like they've either aborted or they weren't fertilized or they've died since. I want to be able to count that because it's important for me to manage my animal welfare and also to prevent mass die-offs later because of a fixable problem. So we're looking at this and we're just like, no, there's just no way we can do this. No. They're really glossy. They have to stay in water, by the way, moving water, because they are, breathing the oxygen from that water. So we developed a photo box called the Row Box, and it actually has a water system that comes through. And we changed the way the light was coming in through a couple of different polarizers, and now the image looks really cool. Now you can actually, with your naked eye, see there's some different phenotypes in here, right? So what we did is then we used that image to then identify and classify the viability status on the trout eggs using a RoboFlow 3.0 object detection method. And then we also kind of compared it to human calls versus predicted calls, and it does pretty good. There are a couple of unfertilized and false positives we're not real good at distinguishing, but we can certainly say, well, these are healthy and these are aborted. And lastly, there's a lot of nematologists working at the USDA, and they do a lot of challenge experiments where they're taking a lot of new varieties of crops or different cultivars and challenging them with different species of nematodes to determine what is the fecundity of that interaction and if there's any native plant resistance in any of the cultivars. But it's really time consuming because you have to make serial dilutions and then you have to sit down a microscope with a clicker and then you're just counting for weeks. 3 people for 3 weeks sitting there. And it's okay, but it's not that great. It's not that accurate. So now they started to do it in microtiter wells and 96-volt plates with a computer imaging cell counting system. And then they just needed the software to do the egg detection. And so we built a piece of software called NemaQuant that automatically will do the egg counting for you. You'll get the masks here so you can go through and double check it and QC it. It has really good repeatability and it's a huge time saving. So they make 2 dilutions, put it all in a plate, run it, and then the data is done by the end of the day. It's also really user friendly. So we don't expect our breeders to know how to code an R or to, put a RCNN mask on algorithm on anything. So we really work with them to say, how much more user-friendly can we make this for you? And the nice thing about this one is it is extensible to other crops. There's, like I mentioned, there's a lot of nematologists out there. This may be the new Blackbird for nematologists because it is something that is easy to deploy and it's already built. You don't have to do any tuning for it. And lastly, we have a number of our berry breeders collect lots of images of berries by the bush, but also by the plot. And so they're taking a lot of high quality images in 2D, but processing them takes time. So we built a really small, clever little tiny app where they can just do batch processing. It's no code solution. So they just upload their folder of images. We've already tuned the pipeline for the species and traits of interest. And then the software extracts the trait data from the photographs into flat CSV files for further analysis. And in terms of breeding decisions, there's just one project I want to talk about, and that's allomate. So our trout breeders have to make mate selections depending on what's available, what's fertile, any given day they want to make a cross. So they have a pool of males and a pool of females, and it's not the best male and females that they really want. It's what they have to make the best of what they have. So this Shiny app allows the breeder to put in a list of candidates that they have, upload their pedigree file, and then they can actually use EBVs to make selections for mate pairs and then predict what the offspring, the estimated value of those offspring are. So this is just a quick way to kind of say, okay, we're just going to fish these males and females out. Once we catch them, we know which ones that need to be crossed to whom. And I mentioned the microhaplotypes that we've been collecting. Well, it's a lot of data to manage. And as I said, we have over 160,000 haplotypes we've discovered, and there's more with every project. So we need a way to manage this and use this data in the breeding programs. So we designed it to store multi-allelic genetic data as microhaplotypes. It has a web front end that allows breeders to use a GUI to explore the database. And it allows breeders to better manage their genetic resources and mine for rare or new alleles and make informed decisions on selection. But this is still in feature build out because there's a lot of different species to cover in this. And also, we work really closely with our breeders to publish with them. Oftentimes, we're the middle authors where we're just doing a lot of the methods or the helping with the analysis. But this year, we've already published or have in various states of publishing 13 publications this year. And we aren't even at the end of the year yet. So this has been valuable both for us, not because it's not one of our metrics, but our breeders feel empowered to do this. A lot of these groups haven't published this much in years, if ever. So this is exciting for them to feel like they have a partner that can help them make more of the data that they're already collecting. And I'm going to leave you with one return on investment story from alfalfa. So alfalfa didn't have a marker panel when we started. And when we started working with the pathologist up at St. Paul, Minnesota, she mentioned aphanomyces is a very deadly disease for alfalfa, and it causes up to 70% crop loss. And alfalfa is a big market. It's $9.5 billion to the farmers in the US. So we did some QTL studies. I mean, this is not rocket science. This is stuff that people have been doing in other species for many years. But we did this in an autopolyploid, and we detected 2 different major QTLs for 2 different pathivars that together provide complete resistance to the fungus. And lastly, I'll talk a little bit about Delta breed. So as we were building out all of our support and all this data that we're generating, we're using a lot of tools under the hood, and it just didn't make sense to share those tools and say, well, first you go here and then you use UpDog and then you go over here and then make sure it goes into GWA. We wanted to put all of this complexity under the hood so that the user, the breeder, is just interacting with one unified system. And we built this all because of BRAPI. So BRAPI is the communication we're using to keep that modular structure. So right now we have we're using GWA as our genotypic data storage, but you could swap it out with anything else as long as it's BRAPI compliant. So this gives us the ability to create platforms that meet the needs of our breeders. It's species agnostic. It allows for growth. It also acts as a connection for other tools. And it's really the hub of the day-to-day breeding program management. Just a couple of features about Delta breed. We use ORCID for authentication, so we don't have to manage anybody's password. Each breeding program is a team, and you can share or privatize your trait ontology as you wish. You can import your germplasm records and capture attributes and passport data along with that data. You can also see that in our germplasm records, we do use UUIDs, universal unique IDs, for all of our data points, but that's really hard for humans to deal with. So we use this GID, which is just a human readable countable number that allows you to have duplicate names in the system. So when you're working in clonal or perennials and you're making cuttings, you can actually have division of that germplasm that now has its own future. We also have a pedigree viewer, and I'll talk a little bit more about that. So the pedigree viewer is actually a BRAP, which is a BRAPI compliant modular piece of software that's dedicated to a specific task or analysis. And in the case of the pedigree viewer, we shamelessly borrowed from BreedBase and made a few tweaks to our fork of it to make it a little bit more useful for our pedigrees, which aren't as deep as many of the other major crops, a lot of unknowns are in our pedigrees because they've only had like 4 or 5 generations. So instead of having one unknown where all the pedigrees tangle, we use a GID of 0, which just means we don't know who it is, but they're not all the same. We also have included over 70 predefined breeding methods across animals, plants, and if that doesn't suit your needs, you can always create one yourself. We also have a prototype of our genotype view using GIGWA as our backend storage. And you can see that in this blueberry example, we're looking under the genotype tab. The chromosomes and the marker information is here, the ref and alternative allele, and then the 4 homologs of the genotypes for just the target SNP are shown here. But we also have a way to display the microhaplotype IDs. We can click on the details panel and then actually look at the pileup of all of the haplotypes at that locus. Our ontology is more than just traits. It can be phenotypes, but it can also be germplasm attributes, passport information. We have an ability to have archive traits. So you have maybe data that you collected with a meter stick, but now you're fancy and you do something else. You can archive that trait. Your data is still safe, but you won't add new data to that older methodology. And our experimental details really try to give you a high level at the top of what is the experimental metadata associated with this experiment? What is the data summary? And then how do I get this data back out when I want to do something with it outside of Delta-Breed? And there's a lot of options here for exporting all the environments together with or without timestamps if they're available, and a number of different formats. And we've been spending the springtime really rolling out Delta-Breed to a lot of our users. And we've got some early adopters already. Most of these groups are using it because they're already using Fieldbook, and the interconnection between Fieldbook and BreedBase is Delta-Breed is very simple to do. And so they find it's really easy to pull their experiments into Fieldbook from Delta-Breed. So I wanted to take one moment to just kind of talk about the lessons we learned along the way. We learned it's extremely important to engage with breeders early, and you really have to listen to their needs to create trust. And that allowed us to triage and tailor support. But we also need to know where they want to go, because a lot of times they'll say, I want to do GS. And you're like, well, do you have markers? And they're like, no. Well, okay, we need to have markers before we do that, or we need to have more phenotypic data. It helps us know what the foundations are needed or missing to get to that end goal. And we found that it's best if we can become a research partner with them that genuinely cares about their success. I mean, this is their livelihood, this is their work, and we are not the breeder. Meeting the breeders where they are and letting them lead us. We don't go in and tell them what their problems are. They come to us and tell us what they need. And there were plenty of people that pretty much slammed the door in our face, and we didn't push too far because it's more likely to alienate somebody, and maybe someday they'll come back. And we don't want anyone to feel like they can't work with us. And we also make sure we convey that working with BI is an invitation. It's not a mandate. You are welcome to use whatever of our services we have, or none of them. And some have chosen that. And we also redefine that when we acknowledge the breeder's success, that's a BI success too. Our success is not separate from theirs. And for the last couple of minutes, I just want to talk a little bit about BI's future at University of Florida. So we're being moved as a unit to University of Florida IFAS in Gainesville sometime before the end of this calendar year. And this is actually good for scientists because it provides more access to BI support for more diverse breeding goals. And so we're expected to now work with any species the USDA ARS breeds for. We are supposed to also support any species the National Arboretum breeds for. And potentially, although not right now, US Forest Service is looking at how they could also leverage some of the tools within BI to do more forest reforestation or preservation of the Forest Service. And it's not just breeding for yield anymore. There's a lot of breeding for different markets, and we have the capabilities and the flexibility to actually tailor the breeding programs to maybe more ecosystem services, or maybe carbon capture, producing diverse proteins, or maybe just even more fuel and polymers. It doesn't have to just be about food or nutrition. And so with this change comes a change in our tech transfer model. So our support at Cornell has really been holistic. We support breeders' needs across many areas simultaneously. But at UF, we're going to probably have to do more project-based. We're going to be able to support more one-off projects, but they're going to be smaller deliverables and shorter timelines. We have also been able to do extensive support at Cornell, from experimental design all the way through to publication. It's going to be a little more piecemeal, I think, when we get there. More small projects with limited scope. Some things we may not go to the publication, or might have to be a separate project because we just can't linger around for another 2 years on a project. And also at Cornell, we've been able to do a lot of discovery-based research and science. We had the freedom to experiment with new technology and have very low risk of it having to produce anything. And I think we're going to be limited more to doing our proven methods, which means less experimentation, but we could deliver more on more projects. And as I mentioned, this team is pretty amazing. Almost every single one of these people wants to come with me to Florida, which brings me to tears all the time. And I've had amazing support from Cornell. I've had amazing support from Ed Buckler and Jean-Luc to keep this project going. People at ARS I've never met who have advocated for us. This is a whole community that's moving, and it's not me, it's everybody else. So I could not do this without the team that has built all these things. I'm just the figurehead that talks about it. So with that, I'll take any questions you might have. Thank you.
This is, in my humble opinion, one of the most strategic and effective uses of federal and taxpayer dollars I have ever seen in the plant sciences. So let's.
Thank you.
All right, questions? From a graduate student first. Perfect.
Are you able to record any of the decisions that breeders make when using your tools, or do you kind of have to infer from like future follow-ups to the data?
Great question. Yeah, there's not a great way to track adoption once it leaves kind of our realm. That's not to say we can't. Like, we have GitHub and a couple of other things that do some tracking. It's really high level though. It's not. I used X for X, so we don't really have a good way of doing that yet. As we build out Delta Breed, maybe more with some more analytics, we might be able to get a little bit more at those numbers under the hood. But for now, it's kind of like they either run with it or we run with it with them and relay it. So I don't really think we have anything that supports that yet.
Did you say you convinced all the breeders to use DART genotyping?
Yes.
I mean, that makes it really easy for you because there's so many other types of.
Absolutely. No, you're right. The other nice thing is a lot of them don't really know what they want or care what they want. But if someone comes to them and says, I made your shirt space very small, this will work. We've got great buy-in. Everyone else is using it and likes it. They're like, that works for me. Let's try it, And it's actually had an effect on other groups too. Like Strawberry built a panel, but they paid for it. Steve Knapp's group paid for those panels, but he decided to build it because of our success working with blueberry and alfalfa using that system. And then Jeff Endelman also built one for potato, a brand new one for potato. And now he's using, so we're doing a lot of collaboration with that Tools for Polyploid group to try to make these multi-allelic data models useful. Yes.
I think two parts of my question. So can you share like why in the first place there was need to start this initiative instead of just working from UVA?
Yeah.
And like Mike pointed out, what do you think are some key points that made this initiative so successful within this short period of time?
Okay. The first question is, why is there even a need for something like BI? And the reason is there are 95 locations at USDA ARS across the country. You cannot build one of these at every location. You might not even have a statistician at any of those locations. So the way our model works is we kind of are providing these micro time commitments to each of the groups, and we have an economy of scale to do that. So while, holistically in the terms of the whole ARS budget, we might look pretty small, we actually provide a lot of services out distributed. And the second part was, what was the second part?
Some key points that you think might have made this initiative great.
What made this, from my perspective, what I think made this so valuable was good steer from Ed and Jean-Luc. And the people that we hired, every single one of them was like a unicorn. Okay. They came in and they're like, you know what? I love research science, but I don't think tenure tracks for me. And I was like, oh my God, I got a job for you. These are the people that I very much identify with. And they wanted to do great science, but didn't want to have to deal with some of the other hassles of an academic appointment. And, and some of them have gone on to do more company jobs. So others have gone on to more academic jobs. It's a place for anybody to learn all of the soft skills, but it's also a great place to still do experimental science. And so I think that hook kept the people that have stayed interested. And the way we've also structured the teams is that nobody is siloed. You may be coming in with genomics experience, but you're having to use it across everything. So you're going to have to learn to go outside your comfort zone. You may be, an expert in sorghum, but guess what? We're going to put you on this other project over here and you're going to have support, but you're going to learn a lot. So there's always this opportunity to expand their expertise. And in the meantime, they're managing projects, they're managing budgets, they're managing time commitments and staff. These are all things that you need at any job you go on to.
Doug, do you have a question?
I have two questions. Just ask one. So I was teaching, so Kim, maybe you said that. I wonder if you have any way of measuring the economic value of what you do, because it's not in the vacuum, right? It's like you work with breeders, and then it's the value will be down the pipeline, but it stems from what you do.
Yeah, it's I spent the last 4 months putting together return on investment slides and stories to try to communicate the amount of money that's gone into here and its propagation of return. It's just so difficult to do that because we're not working directly with a farmer, we're working with breeders. So there isn't, it's like extension for breeders instead of extension for farmers. So it's not really good methods to track that. So a lot of it is just trying to make these, put things together and say, like in the alfalfa example, they weren't able to find those QTLs. Now they can actually use marker-assisted selection in all of their breeding and pre-screen materials for that. That speeds up and brings more crop security to that the U.S. market because of that. So it's trying to tell those stories in a way that are at a 8th grade science level, for the people who are in power. And Congress, they required an 8th grade science level, so it's Congress too. Yes.
I think in its next new home and new iteration, there will be room for someone who crosses that gray line between farmer and breeder.
Absolutely. Yeah. So one of the nice things about IFAS is that it has a very rich and robust extension program, and the stakeholders in Florida are very invested in the University of Florida's extension work. And so I think you'll have a lot more stakeholder engagement ability because there's a lot more horticultural crops down there. And, it's a very agriculturally rich state. So I think we'll find that we get to interact a little bit more with what the farmers are looking for.
Are there any specialty crops that you haven't had the chance to work with that you'd really like to? And also livestock.
Yeah, okay. Livestock, I would really like to work with some of the shellfish. We don't work with shellfish yet, like some mussels or our clams. I think that would be cool because they're pretty interesting. And they have very fast genetic divergence. So they move really fast genetically. The specialty crop I'd really like to work on is mushroom. And the reason I'd like to work on mushrooms is because it provides a challenge that we haven't seen yet. So mushrooms have mating types, and it's a lot more complex than male female. So there's these mating types and they can't work together. So that just makes the complexity of deciding what you're going to use to make your next cross that much more difficult. And we built Delta Breed with that idea in mind, that it could deal with more than just 2 genders, because someday we might be breeding for mushrooms.
I think we have one more question.
Okay.
This is a really interesting talk, especially for us working in specialty crops. It gives, like, it just makes me very, very happy
Good.
on this. And I think as we're going through grad school and working to maybe start our own breeding programs, what do you think that we should keep in mind as we go through, like with knowledge about databases, or what do you think is like something that we should take away and take to maybe our first breeding program?
Yep. Yeah. So I kind of come back to 2 things. One is that you work in a data science now. So you need to be thinking about how do you aggregate your data. And at this point, I don't have a preference for how you do that, but you should have something that works for you. And we've also told breeders, oh, you like Microsoft Access and that's working for you, then we're going to back away, right? You need to make this work for you, but also recognize that there are new tools coming, like AI and other tools, that might make some of the decisions that had to be made over complex algorithms a lot faster. And so being open to new technologies, and also being not afraid to walk away from something that's not working for you.
Moira.
All right. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.